In this video, we will discuss about a technology by which we can do the in vitro analysis of the cell migration, or in other term metastasis. Usually, whenever a lab wants to measure a metastasis property, they need to use an animal model where they can observe the cell of interest or movement of cell of interest from one part of the organ to the uh, another part of the or to the another organ within the animal. So, whereas in this technology we will use a microfluidic device that they have developed and named it as Amica, that can be used to monitor different types of metastasis properties of the cells. So, in this video we will cover how this microfluidic device is prepared, and in subsequent videos, we will cover some of the experimental analysis that they have done with their newly developed Amica. So, in order to prepare this, this microfluidic device, they take a they take two types of uh, plate. First one is the flat plate, and another one is the imprinted gel. Uh, First one is the flat plate on which they develop a PA flat gel. PA is polyacromide gel that they have used, and they developed a flat gel on this flat surface. And for the in in in, addi- in addition to this flat plate, they have also developed this imprinted gel, imprinted PA imprinted gel on this sulfur wafer that helps to form this PA imprinted gel mold that we can see here. And later on, what they did is they combined uh, these two gels, and then observe the characteristic or the force exerted by the cells when they are flow through this kind of gels. That we can say what are the observations that they can do. They can measure. They have measured the 3D single cell migration and also the collective cell migration, and simultaneously they have also measured the force exerted by the cells at the time of migration. So this is the detailed information, detailed figure about how these gels are prepared. So first they have prepared this PA imprinted gel by pouring polyacromide on this silicon wafer with this microchannel design. And after that, after the gel is imprinted, they swell it in the PBS for 1 to 3 days at 37 degree, so that it will absorb the water to its maximum capacity. And similar studies are done on this activated glass cover slip, where what they use this polyacromide based gel on this flat surface to develop this PA flat gel, and they have also kept it in the PBS at 37 degree for 1 day, so that it will it will get its maximum volume. It will absorb PBS and get its maximum volume, and thereafter they blow the air to get rid of the excess PBS that are present on this kind of gels, and then they spread this BS3 on the flat gel, and then place imprinted gel on the top of this flat gel for 1 hour, so that they will form a bond, amide bond. BS3 helps in the formation of amide bond, so that the imprinted gels and the flat gels can submerge to each other. Thereafter, in order to before uh, before directly observing the proteins migration or cell migration, we need to provide a ECM or extracellular matrix on which cells can bind. So in order to provide this extracellular matrix, they need to coat the gels with this SANAP, and that is under UV. And after that, they can put this ECM extracellular matrix and then wash the cells to get rid of the extra ECM, and then they can put the proteins and observe the different analysis of the cell migration, such as what are the force that it exerts. And in this panel B, it shows the different properties that are different properties of this imprinted gel and the flat gel. That is, the swelling rate of this imprinted gel and flat gel varies, and depending on the stiffness of the gels, and the more stiffer the gel, the less swelling will occur. The less stiffer the gel, more swelling will occur. And this observation is observed for both the gels. As we can see there, 8 kilopascal has more swelling rate. And when it comes to the drying rate, and it is very common thing to observe that less volumes will leads less time for drying, and since the flat gel has the less volume compared to the imprinted gel, it will take less time for a drying, as we can see here. And these things are apart from that, they have also measured this young modulus or the stiffness using their uh, they have also measured the young modulus of the gel using some of the tools that they have. I'm not able to recall the exact name, and they can see that they they have made the three different kind of gels. One has around 8 kilopascal, another one has around 15 kilopascal, another and highest stiffness is around 25 kilopascal. And this is the pictorial representation of how this gels looks like. Panel C. So I hope you have understood how this Hemica gels are prepared. In the next video, we will discuss what are the properties that are being observed in order to get the idea about the uh, cell migration of the cells under study without using the animal model. So that's all for this video. Thank you for your time.